Hello everyone. Our discussion is on Module 2, which is plasticity and soil structure. But before we move on to plasticity and soil structure, let us have the learning outcomes. So at the end of this module, the students are expected to define and derive the liquid limit of a soil, define and derive the plastic limit of a soil, define and derive the shrinkage limit for a soil, explain and solve the quiddity index and consistency index, describe the activity of clay minerals, interpret the plasticity chart, describe the soil structure. So basically, Module 2 is on plasticity and soil structure. So when we talk about plasticity, of course, before we have studied the natural components of soil and its various relationships. So now we are on plasticity. So meaning to say, we talk about plasticity of the soil it is defined as the property by virtue of which it undergoes deformation without cracking, fracturing, or rupturing. So plasticity is due to the presence of minerals surrounding the soil solid and water present in absorbed layer of the soil solid. So basically, we talk about for this chapter, when the clay minerals are present in fine-grained soil, the soil can be remolded in the presence of some moisture without crumbling. So this cohesive nature is caused by the absorbed water surroundings the clay particles. So there was a Swedish scientist in the early 1900s. His name is Atterberg. Atterberg developed a method to describe the consistency of fine-grained soil with varying moisture content. So, meaning to say, Atterberg he was a Swedish scientist and he studied about the consistency of fine-grained soil with different moisture contents. So at very low moisture content, soil behave like solid. So of course, at a very low moisture content or at very low water content, the soil will behave as solid. However, if the soil has a high moisture content or water content, the soil and water may flow and it will behave like liquid. So depending on the moisture content, the behavior of the soil can be divided into 4 basic states. So based on Atterberg theory, there are 4 basic state of a soil, which is dependent on the moisture content. We have solid, semi-solid, plastic, and liquid. So we have here an illustration, of course, of the theory of Atterberg, and the theory was called as Atterberg limits. So if we're going to observe from this figure, we have 4 basic states. We have the solid, semi-solid, plastic, and liquid. Okay, this is dependent on the moisture content. So low moisture content, the soil will behave as solid. At a very high moisture content or water content, it will behave as liquid. So the moisture content in percent at which the transition from solid to semi-solid state takes place is being defined as shrinkage limits. So for this one, in this illustration, we have identified now the basic states of the soil dependent on the moisture content. So we have solid, semi-solid, plastic, and liquid. So there is a transition. So as we increase our moisture content, the transition from solid to semi-solid, that is what we call as shrinkage limit or the SL. So again, if we increase the moisture content, the transition from semi-solid to plastic, it is called as plastic limit, which is our PL. If we increase again the moisture content, the transition from plastic to liquid is what we call liquid limit. So these parameters, shrinkage limit, plastic limit, and the liquid limit is known to be as Atterberg limit. So this chapter will describe the procedures to determine the Atterberg limits. So meaning to say, as we go on, in this chapter, we will be discussing how to obtain the liquid limit, plastic limit, and the shrinkage limit. These three are called the Atterberg limits. This was derived from the transition of the state of the soil dependent on the content of the moisture. So we start with the liquid limit. So, okay, basically, what's the importance of the Atterberg limits? So the different Atterberg limits stated above represent the moisture content of the change from friable to plastic consistency. It represents the minimum moisture percentage at which the soil can be paddled. So let's start with liquid limit. So what is basically is the significance of the liquid limit? So the value of the liquid limit is used to classify fine-grained soils. It gives us information regarding the state of consistency of soil on site. The liquid limit of soil can be used to predict the consolidation properties of soil while calculating allowable bearing capacity and settlement of foundation. So basically, liquid limit is important in the prediction for the consolidation properties of soil as we calculate our allowable bearing capacity and the settlement of a certain foundation, which is very useful before the construction of any structure. So for the liquid limit, it is measured using a device. So for the liquid limit, you are going to use a device. This is called the Casagrande device. So you're going to, for the Casagrande device, you have the cup, you have the base, you have the grooving tool, you have the wedge grooving tool, you have the handle. So how are we going to perform? Actually, this is done in the laboratory. How so that we can obtain the value of the liquid limit. So that we can obtain the value of the liquid limit, you're going to place a soil sample inside the cup. Once there is a presence of soil sample in this brass cup, you're going to split at the middle part of the soil sample using a grooving tool. You're going to use this grooving tool to split this middle part of the soil sample. After you have split the middle part of the soil sample, you're going to use the handle and make 360 revolution, meaning to say, as you revolve this one, this will be, the brass cup will goes up and it will goes down. So as it goes up and goes down and it touches the base, that is one revolution or one blow. So you continue your revolution using the handle. So when to stop? You're going to stop once the center of the sample touches each other. So you're going to stop. So you're going to record your number of blows for that particular trial. So that is considered as first trial in the laboratory experiment. So for the standard number of blows, the standard N, because when the soil sample touches, the middle part touches each other, that is, you're going to stop and you have to record the number of N or the number of blows as the soil sample at the center touches each other. So for example, your N is 16. So you have to record that one. First trial, you have N is equal to 16. Then, can you say that the value for, what's this? The value, of course, the soil sample that you place here has a percentage of the moisture content. So you have to record that one. So the moisture content, you record, and the number of blows N as it touches the, middle part of the soil sample. So you have to record moisture content and the number of blows. The number of blows, for example, N is equal to 16. There is, of course, you have a corresponding recorded moisture content. Supposedly, that will represent the liquid limit. However, the standard liquid limit is 25. N is equal to 25, meaning to say the standard N or the number of blows is equal to 25. However, in your first trial, you only obtain N, which is equal to 16. So you have to go again another trial. So second trial, for example, you maneuver this handle, you make revolution, and you count the number of blows as it touches the base. So N on the second trial, for example, is 19. So you have to record, and you have to record also the corresponding moisture content here that the soil sample has. So still that is not 25. Again, you have to make another trial. For example, the third trial, N is equal to 21. So still that is not the standard number of blows. So fourth trial, N was 23. You did not obtain N which is equal to 25. So you have to record, once you have not, after the fourth trial, since you haven't obtained the standard number of blows which is 25, you have to graph that one. So you're going to graph your recorded number of blows in this manner. So meaning to say, a while ago, you have your N, first trial, for example, second trial, third and fourth trial. So you're going to record that one. For example, the first trial, you have 16. On the second trial, you have 21. For example, the third trial, that is approximately 27. On the fourth trial, this is 31. So you have to plot your curve. Of course, in plotting, the number of blows, there is a corresponding moisture content that should be placed on the y-axis. So right, this was 16, then pair up with the moisture content. So you plot that, you place your dot here. Then 22, for example, N is 22, moisture content was 44. So you have this dot, then, and so on and so on. Once you have plotted the dot, you're going to connect the dots. So this is how you connect the dots. And this is what we call as the flow curve. So once you have plotted your flow curve, once you have plotted the dots of which represents the number of blows which corresponding moisture content, you're now going to identify the moisture content, the liquid limit. So how to obtain the liquid limit? As mentioned a while ago, the liquid limit is the standard N is 25. So you locate on this axis 25. So this is 25. So from 25, you draw a line vertical. It should touch the flow curve. Once it touches the flow curve, you draw a line horizontal towards the y-axis. Then after you have drawn that line, you approximately read the value of your moisture content. Approximately, for example, this is 42. So this 42 is the moisture content that you have read for N, which is equal to 25. So that moisture content now represents our liquid limit. So meaning to say, for that particular trial, for that particular experiment, your liquid limit is 42. So you have now obtained the liquid limit. For example, during your trial here, you have obtained on the first trial, this the split here touches each other, and you're lucky that N was 25. So you will not, that would be enough that you will not perform another trial because you have obtained N is equal to 25. So when N is equal to 25 on that first trial, you have to identify the value of the moisture content present. Of course, you have measured the moisture content present. So that corresponds to the first trial wherein you obtain N is equal to 25. That moisture content present wherein your N was equal to 25 during the first trial now is equivalent to the liquid limit. So you're not going to have another trial because you will not have the graph just what we have presented, because you have to obtain now the liquid limit with the number of blows N, which is equal to 25, and that now represents the moisture content for that represents now the liquid limit. For example, the first trial, you have not obtained 25, that was 16. You go with the second trial, and your N now, the second trial was 25. So you will not have the third and fourth trial, you will not have the graph because you have obtained N is equal to 25, and the moisture content corresponding to that is now your liquid limit. So that is the discussion for the performance of the liquid limit.